Hello, we are going to discuss about transcription. Transcription is the one of the important process which takes place in the living system. Before entering into the topic, we may have a look on the word transcription. Transcription is actually a single word, but for a better understanding, we may divide the word into two. So one is trans and another one is script. Trans means copying and the script means written form. So by looking at this, we may assume that we are going to discuss something about copying, writing and processing for something. So the transcription is the process in which a gene's DNA sequences copied or transcribed to make an RNA molecule, and the transcription is generally considered as the key control point of all living system. So who is the central dogma of life? The central dogma of life is DNA. DNA is deoxyribonucleic acid. The section of DNA is called gene. So the gene contains the instructions for making proteins. So when a gene is turned on, the enzyme called RNA polymerase attaches to the start point called promoters. It moves along with the DNA and makes mRNA strand. Based on the codes in DNA, new bases were added to make mRNA. This process is called transcription. So before mRNA used as a template for the production of proteins, it needs to be processed. So this process involves removing of introns and joining of exons to form coding mRNA. So then the messenger RNA moves out of nucleus to reach cytoplasm, where the protein factories called ribosomes in the cytoplasm binds the messenger RNA. The ribosome reaches mRNA which codes to produce triplet amino acids. There are 20 types of amino acids. So the transfer RNA molecules carries the amino acids to the ribosomes. It produces long chain of amino acids using corresponding triplet codes delivered by the transfer RNA. Once the final chain of amino acid is added, the chain falls to produce protein which is coded by the DNA. Here the animation explains the gene which is turned on, the enzyme RNA polymerase joins and moving along with DNA to produce mRNA. So RNA polymerase leaves the DNA at the terminator region. The region between promoter to terminator is called RNA coding sequence. Then the RNA processing takes place to remove introns to make protein coding sequences, and the removal of intron and joining of exon is done through spliceosome. Once all the chain of amino acid is added, then the protein is produced. So finally in the transcription, so these all are the list of components which takes part in the transcription. They are DNA, RNA, mRNA, RNA polymerase, promoters, terminators, introns, exons, spliceosomes, and the final part is protein. So we have discussed about transcription, how they are taking place in the living systems, then what is the output of transcription. So these all are the things we have covered today. Thank you very much.